Hello, everyone, and thank you for attending today's webinar, Assessing Cognition and Driving Ability, Examining Simple and Cognitive Domains in Clinical Pharmacology Studies. I'm Sharon Saltz, Senior Editor of NeuroCentral, and I'll be your host for today's event. Before I introduce you to our speakers, I'd like to quickly cover a few housekeeping items. All the widgets are resizable and movable, so feel free to move them around to get the most out of your desktop space. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right-hand corner. A copy of today's slide deck and our feedback survey are located in the resource list. We encourage you to download any resources or links that you may find useful. Please let us know your thoughts on today's webinar by tweeting us at neuro_central or by using the hashtag NCWebinars, or simply share today's webinar with your friends and colleagues using the Share This widget on your screen. Some networks cause slides to advance more slowly than others, so logging off your VPN is recommended. If your slides are behind, pushing F5 on your keyboard will refresh the page. You can find additional answers to some common technical issues located in the Help widget at the bottom of your screen. An on-demand version of today's webinar will be available approximately 1 hour after the webinar and can be accessed using the same audience link that was sent to you earlier. So, we are delighted to have Gary G. Kay, President at Cognitive Research Corporation, and Ingrid Holmes, Vice President of Global Clinical Operations from Altasciences, presenting for us today. Gary received his PhD in Neuroclinical Psychology from Memphis State University and completed his clinical psychology internship at the U.S. Naval Hospital. He is a diplomat of the American Board of Assessment Psychology and the American Board of Professional Neuropsychology. Gary also serves as a senior neuropsychology consultant to the U.S. Federal Aviation Administration and also serves as a consultant to airline medical departments, military medical organizations, other governmental agencies, and to the pharmaceutical industry. Ingrid's responsibilities include oversight of all clinical pharmacology units with 400 early phase beds, a dedicated driving simulator unit with 10 simulators on site, and purpose-built inhalation facilities. She is also responsible for the harmonization of clinical processes across Altasciences sites and acts as global compliance lead within the quality management system. Ingrid started her career in clinical research in 1995 at Lab Pharmacological Research. Ingrid has extensive experience in early phase trials, international regulatory requirements, business operations, quality management systems, and Lean Six Sigma. We invite the audience to submit any questions you may have for our presenters throughout the webinar using the Q&A widget to the right-hand side of your screen. We will pose your questions to Gary and Ingrid in the live Q&A session after the presentation, and then follow up any outstanding questions offline. Thank you again for joining us today, everyone. I'll now hand over to Gary and Ingrid to start today's presentation.
So thanks, Sharon, for that introduction. I'm Ingrid Holmes. I'm VP of Global ClinOps here at Altasciences, and Dr. Kay and I are very excited to speak with you all today about the topic of the webinar, which is Cognition in Clinical Pharmacology Studies. Just a little bit about our facilities. Altasciences is an early phase clinical research organization with facilities in Montreal, Canada, in Washington State for our preclinical site, and in Kansas City for our other clinical location. We also have a bioanalysis facility and our headquarters located in Laval, which is also just outside of Montreal, and a CDMO location in Philly, in Pennsylvania. CRC, where Gary is located, is in St. Petersburg, Florida, and Cognitive Research Corporation supports trials globally from that location. So CRC and Altasciences have been partnering since 2014. We got together, the first trial we ran was a very large driving trial for a company called Sprout Pharmaceuticals, and we're going to talk to you a little bit about that study later on in the presentation. Since then, Altasciences and CRC have worked together 10 times on driving simulation trials, The vast majority of those have been conducted out of our Montreal facility. After that first study in 2014, it happened to coincide with the release of the draft guidance from the FDA on assessing potential impairment while driving. And we decided that that would be the perfect time to really invest in a dedicated space for driving simulation and cognition assessments. So we currently have 10 permanently installed simulators. These are CRC simulators that are installed at our Altasciences location in Montreal. Each 10 of those simulators are self-contained within soundproof rooms, and the unit itself is completely dedicated to cognition and driving to make sure that we have an appropriate environment to control for any kind of environmental issues, noise, et cetera, that may impact someone's performance during the trial. We can as well, and we have plans to expand to over 20, so should the need for a large, very large driving study come along, we can accommodate that, and so can CRC, which is great. Of those 10 studies, we enrolled over 550 participants. So these are, well-qualified drivers that have been enrolled in the study. And we've conducted over 13,000 simulated drives and CogScreen assessments that go along with them. The studies have had different objectives. So, of course, they're all looking at measuring impairment in cognition. They're looking at, of course, the adverse events that are going along with these potentially new or reformulated drugs, but also there are some differences in design depending on the type of drug we're studying. Some of these are looking at the effects of Cmax and what's going to happen around that time if someone were to get in a car. And others are looking at residual effects. So next morning is generally how that's done. You dose at bedtime or in the evening, and then you're looking at after a night's sleep, what happens to that person if they get in the car to drive to work, let's say. All of that is decided, of course, during the protocol authoring, and the design of the study is decided upon usually based on the planned or thought to be planned dosing, administration, and prescribing information later on once market approval has been granted. We're also looking in some cases to see for newer reformulations of an older drug if they could potentially show less impairment than what's currently on the market. That is a goal of a lot of our clients. And some of those older drugs have not been thoroughly studied for driving trials. And of course, these studies can involve healthy normal volunteers or patients as well. Together, we've looked at mainly CNS indications, schizophrenia, ADHD, depression, epilepsy, and insomnia. But of course, the technology can be used pretty much for any indication. Really, the goal is to assess that impairment potential for these drugs. So a brief agenda for today. The topics we'd like to cover with you all really are to go over the tools for cognitive testing, both on the driving side, but also regular cognition assessments that can be embedded within your early phase clinical trials using the CRC technology. We'd also like to examine the FDA guidance. This is a dedicated driving and impairment assessment guidance that was issued and finalized in 2017. So we'd like to take a look at that and see what changes may or may not be implemented since then based on both the guidance but also just norms within the research as things progress. And then we have a couple of case examples and strategies to talk through with you all, and we'd like to go over the keys for a successful program. After that, we'll also field some questions, so please feel free to load those in at the end of the presentation. I'll pass it over to Dr. K to start on the next slide.
Well, thank you, Ingrid. Thanks for the opportunity to share some of our experience with doing cognitive and driving studies in support of these various clinical trials. And as indicated here, we will begin with a discussion about cognitive testing. So let's begin with the CogScreen test. I'm the author of this test. It was originally developed in the late 1980s as a DOS program for the U.S. Federal Aviation Administration. So its birthplace is in aviation medicine, where really high demands for medical decision-making regarding whether a pilot can return to cockpit following head trauma or if taking antidepressant medication. And this test is used globally, and it's been used not just in medical decision-making, but also in clinical trials. So this is a computer-administered test. We have normative data on over 1,400 healthy adults from age 18 to 85. The new version of this test actually is on a Microsoft Surface Pro tablet. It actually has been proven to provide us with high resolution for timing, millisecond level of timing, and we have over a 30-year history of use in regulatory trials with the CogScreen test. It actually has found its way into decisions about whether a drug might be found to be non-sedating or sedating, for example. The other tool that we use in testing performance and cognition is our driving simulator. The CRC customized driving simulator is the CRCDS MiniSim. And this is a driving simulator that finds its origin at the University of Iowa at the National Advanced Driving Simulator, where they have a full motion, it's a vehicle under a dome that actually moves. That same software was ported for use on a PC, and that is the MiniSim. And we have been using the MiniSim in numerous clinical trials. We have validated it. Its sensitivity has been shown across many different drugs and treatments where we've been able to show both improvement or impairment of driving performance. And the use of this simulator allows for multi-center clinical trials. But the one center we've done the most studies has been with Altasciences, where we've come in and validated all of the instruments and trained and certified the raters who do an excellent job of actually administering and running the simulator during these trials. As I said on the previous slide, CogScreen has been around since 1988 and is used globally. It has many different languages, and the test has a rather exceptional demonstration of sensitivity to drug effects. And CogScreen has been used to measure changes in a range of different cognitive domains, particularly in the areas of attention and processing speed, but also memory, visual perceptual functioning, sequencing ability, logical problem solving, psychomotor speed and coordination. One thing unique perhaps to CogScreen is the measurement of divided attention or multitasking, and also assessment of executive functioning. As I stated, CogScreen has a long history of use both in clinical applications and in clinical research. It's been used with a number of patient populations, as shown here on the slide, and some of these have been for clinical decision-making and others for clinical research. It's also been used to look at the effect of a wide range of medications, as you see on the right side here of the slide, including medications which actually are more CNS stimulants like the amphetamines or CNS depressants, pain medications, antihistamines, a wide range of drugs that can potentially affect CNS functioning. On this slide, you see the abstract and first page of publication for the clinical trial that was the initiation of CogScreen into pharmaceutical clinical trials. And this was our work on Loratadine, a drug you know as Claritin. And this was one of the pivotal studies for demonstrating that Loratadine, Claritin, was non-sedating, showing no difference between Loratadine and placebo, whereas the positive control diphenhydramine, which you know of as Benadryl and is present in many different drugs, caused impairment on the CogScreen measures of cognitive functioning. As another example, CogScreen has been used multiple times to investigate the effects of various antimuscarinic agents to determine the extent to which they were blocking cholinergic activity in the brain and having effects on memory in older adults, and was quite successful at differentiating the various antimuscarinic agents used for bladder control in terms of those which had an effect or did not have an effect on memory or other cognitive functions. And now we switch gears from talking about computer-based cognitive testing to talking about our driving simulator, the CRCDS, the Cognitive Research Corporation Driving Simulator, also referred to as the MiniSim. This is a fully standardized, validated, PC-based driving simulator, which we have worked very hard to optimize for pharmaceutical research. As I'm fond of saying, it is ready for prime time in research to be submitted to regulatory agencies for review. We incorporate the MiniSim, as I mentioned before, developed at the University of Iowa for the National Advanced Driving Simulator, and which they utilized in the full motion NADS-1 simulator that was originally developed for the National Highway Traffic Safety Administration, NHTSA. Before we go on, I'd like to share with you a video that will show you the MiniSim simulator, one of the MiniSim simulators that's presently at Altasciences in Montreal.
At Altasciences, we run the gamut of clinical services. We are the premier service provider in driving simulator studies. So not only can we go ahead and recruit these subjects very quickly based on a large recruitment database, but certainly we can perform these driving simulator studies very quickly. So clinical conduct, recruiting and screening can be started up and then completed within 2 to 3 months, which is really unparalleled. Every single study has protocol-specific training, and that is provided by our partners at Cognitive Research Corporation. Our strategic advantage here is that we have a large recruitment database, which allows us really to triage through the study requirements and identify subjects who may potentially be eligible to participate in driving simulator studies. In addition to the standard screening requirements to identify healthy volunteers, physical examinations, laboratory assessments, there are specific driving simulator tests to ensure that subjects can actually undergo a driving simulator test without any type of side effects such as simulator sickness. We have a long-standing relationship with Cognitive Research Corporation. The expert in the field, Dr. K from Cognitive Research Corporation, visits us prior to each study within 2 to 3 weeks to ensure the training of all of our staff. Several years ago, we were fortunate enough to run a study with a molecule called flibanserin, which was ultimately marketed and approved. Through our partnership with Cognitive Research Corporation, they were impressed with our professionalism, with our clinical conduct, and certainly with the data outputs, which made them come back, like many of the partnerships that we've developed with pharmaceutical companies over the years.
to the use of driving simulators to conduct studies to look at the impact of drugs on driving performance, studies were conducted using what is referred to as the over-the-road method. Basically, an instrumented vehicle is taken on the highway to drive 50 kilometers in one direction, crossing over to the other side of the highway and returning 50 kilometers for a total 100-kilometer ride, where the driver is accompanied by somebody who monitors their performance, can end the trial if necessary, and the vehicle is designed to measure basically the extent to which there is movement of the vehicle relative to the road marking, the white line in the road. And that's referred to on this slide as over the road, or OTR. Well, the FDA has come to a decision that either driving simulation or over-the-road method is acceptable in evaluating the effect of drugs on driving performance in those studies where the FDA has required a dedicated driving study. So the key thing is that whether you are using the over-the-road method or a driving simulator such as as our MiniSim, we're basically going to focus on lane position. Now, with the over-the-road, the rate of data acquisition is about, let's say, 12 frames per second. We are able to record at 60 frames per second using the simulator, so we have a more precise and sensitive measure. But there are other differences, and Ingrid and I commonly encounter questions from sponsors about, well, what are really the differences between the driving simulator versus over the road? The simulator is designed to create basically a feeling of really being in a vehicle. So the simulator, as you saw in the video, uses a full-size cockpit driving seat from a vehicle, from a true vehicle, full-size steering wheel, pedals. The wheel vibrates in response to the road, and the screens represent basically the size of a windshield. So we do everything to try and generate as much as possible the real experience of being in a true vehicle. And the studies really bear out the fact that we find very comparable results whether you use the over-the-road method, the OTR, or using the driving simulator. There's a study where the same drug was evaluated, and while absolute number of centimeters of deviation will vary, the relative effect is very comparable, and results therefore are considered comparable, though one could certainly claim that the simulator is more sensitive. Both have been accepted for regulatory submission, but you do get better experimental control with the driving simulator, where you have control over traffic, over weather, and you don't lose a substantial portion of the data from when in the OTR where they can't visualize the white line in the road for various reasons, and that data is just lost. We don't have that situation with the driving simulator, and we certainly don't have any risk of harm to subject or to other operators in a driving simulator. No subjects have been harmed in our driving simulator. So that's some of the basic things, and be happy to answer other questions. Now, as you see in this slide, and we're really trying to point out that what we're trying to do is create an immersive, realistic environment for the subject, for the participant, in the study. So again, you notice the full-size steering wheel, the pedals, the automotive seat, the vibration you can't see, but is built into how that wheel feels. Audio, we use the surround sound type audio so that you will hear the roadway. And we actually acquire through cameras, facing the driver and facing over the driver's shoulder, we can actually observe the subject's reaction and throughout a drive, which also provides a very nice QC to look at data when necessary. On this slide, I'm trying to share with you some of the keys to making the CRCDS MiniSim ready for prime time in clinical trial research. So that involved developing scenarios, so a scenario for looking at sedation or inattentiveness, executive function problems, developing data reduction methods that generate the endpoints that are necessary for looking at the impact on driving. The data acquisition I mentioned before is 60 frames per second. The development of validation scripts for the equipment or the installation. Any change to any software, hardware, whatever goes through a full installation verification process. There's system performance testing, scenario-based testing. We validate all of the data reduction and the data transfer methods. And all of these are a key to meeting regulatory requirements. Moving on to additional issues in using the CRCDS MiniSim in clinical trials was to demonstrate the sensitivity of the simulator and scenarios to low-dose alcohol. So we have established and replicated the sensitivity to a 0.05% blood alcohol level, which is often a criteria used by a regulatory agency in making a determination about whether or not impairment is present. There's been a study that compared, as we mentioned previously, over the road to next day residual impairment from a sleep medication. I'd say one of the things that I may have failed to mention on that prior slide is while the over-the-road typically is one vehicle at one site in Europe, we are able, as Ingrid pointed out, to conduct at least 10 driving tests at one time at one good phase 1 facility by using the driving simulator. We basically have demonstrated the sensitivity of the CRCDS MiniSim to various CNS depressants, to CNS stimulants, and to various disease conditions that are associated with impaired driving. As Ingrid mentioned, we're planning to share with you just some highlights from the FDA guidance on assessing drug-impaired driving. I had the opportunity to serve as a consultant and still serve as a consultant to the National Highway Transportation Safety Administration on issues related to driving safety and evaluating drug-impaired driving. And some of that earlier work that I did is included and cited in the FDA guidance. The FDA guidance on evaluating drug effects on the ability to operate a motor vehicle was finalized in 2017. They basically point out that they recognize drug-impaired driving as a public health priority, and the guidance was developed to help identify drugs that increase risk of motor vehicle accidents. They point out that patient self-perception, whether you feel you're safe to drive or not, is not really sufficient information for making a determination that a drug does or does not cause impairment. often there's a lack of correspondence between what you perceive and how you actually can perform. The FDA basically pointed out that objective information was needed about how a drug impacts driving, and this is really needed for people to make proper decisions and to enable safe use. The guidance also addresses the importance of first-in-human early development trends that are informative about the potential need for a later driving study for a particular compound. The agency guidance recommends that cognitive assessment be done early in the development process. Tools such as CogScreen measure performance with very good reliability and validity. One can also do assessment using self-report measures such as questionnaires and visual analog scales, which these days can be administered on the tablet, like the CogScreen tablet, or one can also have results from various physiologic measures like quantitative EEG that might be informing that a CNS depressant effect is present. The key thing is to look whether it's coming out from self-report AEs or it's coming out from specific testing to find that there may be cognitive impairment indicating a potential need for a driving study.
And Gary, I would just add to that, what we see quite often now, especially in early phase programs, and this is probably more true now than it would have been before this guidance has been sort of well entrenched, is clients coming to us both looking at their early phase trials and making a decision as to whether they should add in real cognition endpoints to that first-in-human, let's say, SAD-MAD study. In a lot of cases, we still have clients that are not going there, even though the guidance does recommend to do some form of assessment in early phase trials, whether or not you believe you're, going forward with a drug or a compound that will affect cognition or not. They do recommend that you assess it. Most of our clients, unless they're in the CNS space, are still not applying that. But what we do see is when they know or when they're expecting to have an effect, they're starting to think about what they can do much earlier on in their program to help inform and design their later phase work. So in the past, we would have waited, let's say, until the end of phase 2 meeting to make a decision on what has been seen and, is a dedicated driving trial required and all of that. What we're seeing now is clients actually looking at conducting driving or cognition much earlier, so that they can take that information and build it into their phase 2 programs as either a de-risking tool or even as part of the design itself within that phase 2 program. So that's a big difference that I think this guidance has actually influenced.
Yeah, thank you for that point, Ingrid. And based on that, a key question for sponsors is, does my drug need to be evaluated with a driving study? The guidance does address that, gives you some good information. For example, it indicates that drugs that are intended for chronic outpatient use by adults who drive are those drugs that need to be studied. In addition to sedating drugs, because a lot of folks kind of had the idea that maybe this only applied to sleep medications, which is where a lot of the early studies were conducted looking at drug-impaired driving. The guidance actually also applies to drugs that are CNS stimulants because these drugs may have an effect on risk-taking, which would be adverse to driving. The guidance makes it clear that if you have a drug that was previously approved, but is now being proposed for either a different indication or a different dose or a different dosing schedule, or to be used in a new patient population for which there's insufficient information about how the drug could affect driving. In all of those cases, they're saying that a dedicated driving study is to be conducted. This slide addresses some of the key methodological design considerations. Who are you going to enroll in this dedicated driving study? The guidance indicates that they should be patients who are from the population who would likely use the drug. If it's going to be used by older adults, then in fact, they should be included in the trial. And if there's one area we've seen pushback with protocol review, it's been studies that did not include older adults. And so we are including older adults in these trials now. And the question always seems to come up is whether we have to do this in patients who have the clinical condition for the drug, or can the testing be done in healthy volunteers? And generally, the key thing is that if the condition, the disorder, is not expected to have any effect on driving, then in fact it may be appropriate to test healthy volunteers. But one should look carefully at that question in deciding whether use of healthy volunteers is appropriate. Basically, the agency recommends taking a look at both initial dosing, so when you first start taking the medication, what is going to be the effect, and based on how the drug accumulates, or if it's a drug that will have less effect over time, you'd want to look at the effect of chronic exposure. A key thing is they want testing conducted at where the person is likely to have maximal exposure to the parent compound or active metabolite. So dosing time needs to, time after dose before driving begins, needs to be carefully decided upon for designing the trial. That information can identify what the time course is of potential impairment and the extent to which tolerance develops with repeated use. And again, based on the impact of various metabolites and differences in how people metabolize, they are keen on having one look at the highest exposure expected, and in some cases this will involve use of a non therapeutic dose, like a double the dose, even if that's not going to be a therapeutic dose. They may have you include that in your clinical trial. Continuing with the methodological issues on this slide, one thing is that one should include an appropriate positive control, something that's reasonably impairing but is generally from the same class. It doesn't have some kind of extreme effect, but this is to demonstrate assay sensitivity and also to help us understand the magnitude and duration of impairment. Time of day, duration of effect should be well documented. For those who aren't familiar, these are generally crossover designs that are employed. So the patients basically will come in for multiple treatment periods and be tested with the therapeutic compound, the investigational drug, with positive control, and with placebo, and maybe with a super therapeutic dose of the investigational compound. The design is really going to be dependent upon what questions need to be answered. If we're simply looking, like the last study we did at Altasciences, at looking at residual effects, then we limited our testing to looking at next day, next morning residual effects. So we looked at performance the morning following an evening dose. If there's a concern of a drug that's going to be taken throughout a day, then we look at daytime dosing at the onset and duration, or if it's a drug that's being taken chronically. Another clinical report I was working on yesterday, we're looking at the effect both of accumulation but also looking at tolerance for the drug. So this will really impact when you have the drives conducted and how many drives will you look at in that particular trial. Well, a slide that I like to share with you is the following, which shows the effect on what is the primary endpoint in whether you're doing an over-the-road OTR or a driving simulator study. Is typically this thing called SDLP. As it says here, summary of SDLP change from placebo. SDLP is the standard deviation of lateral position. To put that in plain English, it's weaving. How much are you deviating from side to side laterally as you head down the road? So it's a measure of within individual variability, and it is one of the most sensitive indicators to impairment. And so looking at this slide, you see drugs that actually, if it's minus, like the one at top, Plebanserin, actually you get a decrease in the amount of weaving. That's in centimeters. It did not cause any increase. You go down to drugs like Tolperisone, and that drug basically 0.2, no effect relative to placebos. Similarly, gabapentin, negligible, almost no effect. And then you start getting into mild effects. Diphenhydramine after a night of sleep is having a small effect. And then you get into Zopiclone, a very commonly used sleep medication, and even after 8 hours is still having an effect of 3.5 centimeters. And you look there at the confidence interval, you're seeing the upper end of that 95% confidence interval, 4.9 centimeters, exceeds the level that you see right below for alcohol, ETOH, at 0.05, which is 4.4. And so that, is that 0.05 alcohol, 4.4 that we have been able to replicate, really is what the Food and Drug Administration considers to be evidence of impairment. And that's partly based on epidemiological data, which indicates that risk of a crash significantly increases at that level, at the 0.05. And so that is often our non-inferiority limit. We look at the 95% confidence interval for a drug and see if it exceeds that 4.4 centimeters you see for alcohol, 0.05. And then you can look at the skeletal muscle relaxant, cyclobenzaprine, its effect, and then various benzodiazepines at different times and different doses. So this just shows some of the studies and some of the drugs that we are able to share with you on the effect that we see on the CRC MiniSim.
So thanks, Gary. That was a great summary of both the methodology used behind these studies, the guidance, and also the technology that we employ. We thought it would be nice to transition into some case studies. Mentioned earlier that Altasciences has run 10 studies now with CRC dedicated to driving and cognition assessments. We've got a couple of case studies to go over that I think you'll find interesting, that have applied different methodologies for different reasons. In some cases, it has been, purely based on what the FDA has requested of the company in order to demonstrate a safe product for their label claim. But also, there are some other strategies employed by sponsors to take a look at impairment over time, even competitive labeling. So we thought we'd take a kind of snapshot of a few case studies and walk you through what was done and what the results were. So I mentioned earlier that CRC and Altasciences came together with a sort of a first study. Probably it was conducted in 2015, so we started talking in 2014 about this trial. And this case study is actually related to that trial. This was a company called Sprout at the time, and they were studying fulbanserin, which was a repurposed product that was initially being studied by BMS for depression, and had been repurposed by Sprout for libido. So I'll pass this over to Dr. K to talk about the design and what the results were.
Thank you. Ingrid, this was a study to look at residual next day effect. The company decided to look at nighttime dosing of the drug, and they were to carry this out in patients who were of the age and similar patients to those who would be taking this medication as a therapeutic. So as you see, there were 72 healthy premenopausal females between 18 and 50 years of age. Mean age was 48 years. And the study was conducted very quickly, as you see here. From May 12th was our first consent, and the last study visit was September 16th. So this was done with great efficiency and really helped move this product to its eventual approval. And certainly something that I think you would not see being performed at nearly that rate if you weren't doing using driving simulators. So here's a little detail about that study. It's a good case example. So this was done solely at the Altasciences facility, randomized, double-blind, placebo-controlled, Latin square, and the 4 periods, which I'll discuss in a moment. These are healthy women, 18, 50 years of age. And so 2 of the periods are flibanserin, the investigational drug, at 100, and then at a supratherapeutic dose, 200 milligrams. The positive control, zopiclone, and matching placebo. And zopiclone was selected as positive control for assay sensitivity. And as you saw from the prior slide that I showed, the effect of zopiclone is significant relative to the effects of alcohol, 0.05, but mildly. It's not a drug hitting anybody over the head with a hammer. We looked at acute dose effect, but we also looked at the effect after 7 days of dosing. So we did both. After looking at chronic dose, then subjects would undergo a 7-day washout period and then would be crossed over to the next treatment arm. They would dose the medication at bedtime. Subjects were then awakened after 7 hours of sleep, and then they would begin the 100-kilometer, approximately 1 hour driving task. The driving task began approximately 9 hours post-dose. This schematic shows the study design, which is pretty common to studies where we're looking at residual next day effects. So patient administered acute dose at bedtime. Then 9 hours later, they do the driving task. And then they continue with dosing. If this is looking at chronic effect, often we don't need to look at chronic effect, but in this case we did. And then we do another driving assessment on 7 days of dosing. Patient is discharged from clinical site and comes back after a 1-week washout to begin the next treatment arm. And as I shared before, this study had 4 treatment arms. Well, this slide is going to share with you the basic findings. We're looking here at the change relative to placebo in the standard deviation of lateral position, the primary endpoint, again, the measure of weaving. And so basically higher scores are bad here. They mean more weaving. A score below the 0 means better driving, less weaving. And what we see is those little boxes here above the 0, those are both for zopiclone. You're seeing the result for day 2, and you're seeing the result for day 6. So very consistent finding, and you're seeing that that upper limit of the confidence interval does exceed. If we had a little line at 4.4, it's worse than the 4.4. So that's clearly having an impairing effect for our positive control. What that demonstrates for us is assay sensitivity. Now, looking below the 0, you see at day 2, the flibanserin 100 milligram actually is better than 0. So they drove a little better. They slept well that night. And then you look at day 8 after 7 days of dosing, and whether we looked at the 100 milligram dose, the little circle, or the triangle, the 200 milligram dose, we saw no adverse effect on driving performance. Conclusion of the study was that bedtime administration of flibanserin, even at doses up to 200, which is not the super therapeutic dose, does not impair next day cognitive function, which we tested. I did share those results with you. That was using the CogScreen digit symbol test. We did not see an effect for flibanserin. We clearly saw impairment with the zopiclone, but we did not for flibanserin on either the cognitive test or on driving performance. Basically, the FDA review came back with the division comment, the negative findings of next day driving impairment study are reassuring, something that was good news to the sponsor and reassured the agency. And that reassurance is well reflected in the label. You actually find in the package insert a description of the driving study, of the dedicated driving study will now be entered into your package insert, and you're seeing here that what the FDA allowed was in a randomized, placebo-controlled, 4-way crossover in 83 healthy premenopausal female subjects. No adverse effect was detected on measures of driving performance itself or psychomotor performance thought to be important for driving performance when assessed 9 hours following single and multiple doses of ADDI 100 once daily at bedtime or single dose of ADDI 200 milligram at bedtime. So that was all very good news for the sponsor. Well, I'm going to take a minute more to share with you a couple of other studies that have recently been published and where I think they really demonstrate some of the other designs and questions that can be addressed with driving simulator studies. So this study was one looking at a skeletal muscle relaxant, Tolperisone, which I believe is a drug available in certain parts of Europe now, but this was for submission to the U.S. Food and Drug Administration. They compared Tolperisone to a commonly used skeletal muscle relaxant known as cyclobenzaprine. You may know of it as Flexeril, and comparison to placebo. And this study we conducted at 2 sites. We had 35 healthy adults, 22 to 54 years of age. And this study was to look, as it says in the title, at both next day, at acute, and steady state effects. All of that was designed into the trial. So what this study allowed us to do was look, as I said, at acute effect, residual effect, and steady state effect. We studied Tolperisone, the investigational drug, which is a TID drug, compared that to cyclobenzaprine, was the active positive control, and to placebo. For dosing on day 1, we're looking at the acute effect. We then test people after a night of sleep, after their nighttime dose on day 1, but before their dosing on day 2 to look at residual effect. And then we test on day 3 at steady state. And what we found, as you see in the lower part of the slide here, is that under all these Tolperisone conditions, SDLP showed no impairment. And that's done by comparison of SDLP to placebo, by looking at the 95% confidence interval to see if it exceeds the effect of a 0.05% blood alcohol, and by doing symmetry analysis to look to see if the distribution of the paired differences from placebo are symmetrical about 0 or not. And the FDA really demands that all 3 of those analyses be conducted to demonstrate non-impairment. What I'm wanting to share with you on this slide here is really the result for the positive control for the cyclobenzaprine. And what you see on the left side is that little dotted line is at the 4.4 centimeter, the 0.05 blood alcohol. You look at the number of subjects driving with cyclobenzaprine, very few of whom actually thought that their driving was affected. They felt they were safe to drive, yet they were impaired. This is really reflecting that lack of awareness of impairment, which is why these dedicated driving studies are so important. The other thing I'm showing on the right side of the slide is that there is some learning to drive under the influence that's taking place from day 1 to day 2 to day 3 if you look at the gray bars for cyclobenzaprine. The last case example we're going to share with you today is a study recently published on Lasmiditan. This is a drug for treatment of migraine, and we looked at the impact of dosing either at 1.5 hours in a first study and then at 8 hours in a second study. And while impairment was found at 1.5 hours post-dose, there was no impairment shown at 8 hours post-dose. Very important information for both people who are going to use the medication and for prescribers who will be telling people about when it's safe to drive after taking the medication.
Great, thanks, Gary. So, just to conclude here, in summary, we hope those case studies really demonstrated that there are multiple methodologies that can be employed for cognition and driving assessments. Really, it's going to depend on both the qualities of the drug we're studying in the patient population it's intended for, but also potentially the strategy of the sponsor as well, and what the current state of market authorization for similar products is. So you really can see that there are different ways to study these things, and how each can be quite important in the final product, which is your label. And then, of course, just to remind everyone that Altasciences and CRC, I'm sure you can see from the presentation, has a very strong relationship. We partner not just on the functional deliverables of a program, but on all of the supporting elements as well. So when it comes to how to design the protocol, of course, Altasciences has our own area of expertise, which is the conduct of the clinical trial, the scientific management behind the medical writing and the results. And then CRC has their side of expertise, which of course Dr. K has very well explained and demonstrated their ability to support and to actually define and describe those elements within both the protocol and CSR for your trials. So in terms of key considerations for success, we wanted to just touch briefly on how we're generally interacting with our clients, and what we've seen in the past that has worked really well, and other things that maybe haven't been worked out so well, and why. Really, the main thing for on both of our sides is to start those strategic discussions early as you can within the program. So if you are thinking about repurposing an existing drug into a new formulation, or if you're working on an NDA and it's completely novel, thinking about what you're going to need to do within those early phase studies really, really is key, because it cannot just help you out in the future, but it can also eliminate risks within the program. It's very important to start those discussions early, and we're used to having those discussions with our sponsors regularly. So this isn't something you need a contract for. You bring in people that can help you kick things off, give you some ideas, and then move, through the discussions with people that can help along the way. The other thing that's really important is the relationship between our site staff and the vendors that we use. We obviously at Altasciences consider Dr. K and his team as partners. They feel like colleagues when they come on site or when they're helping us set up for a program. And it actually doesn't matter who contracts who. Half of the studies that we've run together, CRC has contracted Altasciences for, and the other half has been the other way around. The sponsors have come in through Altasciences, and we've subcontracted to CRC. The relationship is really, really important. It allows us to make sure that we have an honest discussion about, how the teams are producing their results. It makes sure that we have well-qualified associates working on all of these assessments, which is really, to the next bullet point, the key when you're choosing a site. And having a team like Dr. K's, where they provide really in-depth certification and training for our staff prior to each one of these studies, ensures that we get the feedback, immediately as to whether our team is functioning the way they should. You need to choose a site that can demonstrate to you that they've done this and done this well in the past. Not every CRO understands why PD assessments are so important. Some of them are actually very simple to execute, such as a simple VAS scale, let's say, but they have incredibly important impact on the results of a study. And if those team members don't understand that even that simple assessment is, linked to the primary objective of a trial and how that program moves forward and the safety of those patients, that they will make mistakes and they won't understand the impact they're having. So this is really, really key. In terms of participants, this is also exceptionally important for studies where we're focusing on PD. In terms of driving, you need to have a database of good drivers, people who actually have a good driving history, who also are willing to participate in clinical trials, will comply with all of the rules we put in place, and that they're reliable and are going to complete that study if they can. There are locations where that's difficult to find. And it really is important to make sure that that's assessed going into it. Looking as well at, we call them good raters, but, study participants who can be trained well and understand, just like our staff, the importance of those assessments, and that they really do understand the difference between one end of the VAS scale to another. It's very important that they understand those things so that they're actually giving their best effort. In terms of driving, you've got a location where you can conduct these studies, but is that location adequate and free of distraction? Very important. At Altasciences, I mentioned we have a 10 simulator suite. All of those simulators are self-contained. They're all held in soundproof rooms and completely free of distraction, and that would be something to look for no matter what type of PD assessment you're going to be working on. That's very, very important. And then the validation of that installation, Dr. K mentioned that these simulators and CogScreen are fully validated, but then what happens when they get on site and who's supporting it? You need to plan for all of that. And then finally, I would say as quickly as we can look at results, the better. This doesn't necessarily have to be a final analysis where, you've got to be careful with blinding and everything else, but you need to know as you're going forward that if something is going wrong, you need to correct for it. So taking the steps to have good observation on the conduct of the trial, both through monitoring, but also the data that's coming out of it is the key.
Thank you for that very insightful presentation, Gary and Ingrid. I hope our audience found it of great interest and value. This now completes our 1-hour presentation, but you are welcome to join us for our Q&A section now, where we will answer some audience questions submitted so far. If you do have any questions for Gary and Ingrid, then please do continue to submit them using the Q&A tool in the right-hand side of your screen. We have received a number of questions so far, so we will address some of these now. Question number 1. As a sponsor, why would I want to add cognition testing to my first-in-human program?
I'll take that one. I believe that you would want to know as early as possible if there are any meaningful changes in cognitive function associated with the compound, whether those are enhancing cognition, which would be great news, or if there is some impairing effect, which you can mitigate in some way by dosing, by time of dosing, in various ways. So I think to learn this early on helps you in your development program.
That's great. Thanks, Gary. On to question 2. What are the main operational challenges the CRO/site sees when conducting a dedicated driving trial?
Yeah, I can take that one. I would say the number one operational challenge, besides timing, because most of these studies, as everyone knows, we're always in a rush, you know, particularly for NDA enabling type trials. But the number one challenge really is trying to balance out the assessments. So let's say this is a driving trial with only one drive. So that's not the most common type of trial we see, but it is the most simple. You need to look at when those drives need to be performed and how many participants are actually expected to make it through to completion on that trial in order to assess these results. It sounds quite simple, but the simulators themselves are going to be rate limiting. Generally with these studies, you want to try and limit your dose time, particularly, of course, if you're doing a next day residual effect study. But you're wanting to limit the time of day that those subjects are on the simulators to a certain window so that you don't actually have a variable introduced by allowing people to be, all over the place throughout the day, which means that you can only have a certain number of people passing through the unit on any given day. And in our case, right now, we've got, 10 simulators. That really would be the maximum group size that we would handle because we don't want to double up our participants, throughout the day on a simulator. We want it dedicated to one subject, one main critical assessment, and that's a rate limiter, and it's a challenge to operationalize on large trials. Particularly if you're looking at a site that only has the ability to bring in one simulator, these studies can go on for a very long time. But we found that 10 is a pretty good number, allows us to operationalize well, and also keep the speed going for the trial.
Perfect. Thank you, Ingrid. The next question says, my company is currently in phase 2 development of a novel antidepressant. How do we find out if and when we need to perform a driving study? And when do most companies make that decision?
Well, I guess I'll take that one. And I would say that basically it's around the time that you're mentioning. You're currently in phase 2, so that's a good time. You're going to want to look at your phase 1 results and see what the adverse event profile looks like. Excuse me, the extent to which you're seeing, people having AEs of a CNS nature, and I think you're going to want to know about, for that class of drug, for that type, even though you say it's novel, what do we know about this type of antidepressant? If in that class we know that there's likely to be cognitive or psychomotor performance effects, then it's more likely that you should be planning for that driving study. And we do those studies often in phase 2, or there may be something that you're conducting while you're also conducting your phase 3 trial.
Perfect. The next question includes, if we are repurposing a drug, changing the formulation slightly, would we still need to perform a dedicated study? Could we opt to use the current labeling or the marketed product?
Well, I would hope that that's clear from the guidance. To some extent, the guidance is saying pretty clearly that if you are repurposing a drug, and depending on how slight that change is, right, with the formulation, that is probably going to be the determining factor about whether or not a study might be needed. We have certainly worked on projects where it has been a formulation change actually with an intent of having an impact upon the pharmacokinetics of the drug to an extent that a driving study was needed. If the formulation, as you say, is slight, it may be the case that it won't be necessary, and that's a good question to basically review with the agency, or certainly we're happy to look at this with you in detail as well.
Yeah, and I would just add to that, there are some strategic reasons to take a look at running the trial as well. If you're looking at a very old drug, chances are they weren't assessing driving at all. They may have a quite severe label associated with that drug based on what's known right now, but that isn't based on an actual trial. So you may have an opportunity to gain an advantage on the existing label by performing a driving study with the right controls and actually have a competitive advantage in the future with a product that's quite similar to something that's already on the market. So that is something that we've seen, and I think it's a good thing to consider.
Great. Our next question says, exactly what data is being collected and analyzed in early phase 1 studies for cognition? How do you ensure good quality of the data?
Well, you're certainly have always been collecting adverse event data, which is informative. But specifically to look at cognition, there are a number of tools, actually computer-based or tablet-based tools that can be quite helpful in assessing and screening for changes in cognition. The ones that you should be using in a clinical trial are those that have a really good history and of demonstrating that they are, following 21 CFR 11. That they are meeting kind of regulatory requirements, that they have, current data demonstrating their reliability and their validity, and that they have really solid data handling, data transfer methodology, included. Those are all really key. I mean, I think that if you talk to raters these days at sites, they're very frustrated by some of the tablet-based systems that are out there that really are questionable. But I think if you select the right material, it can be really helpful for you to gather in a highly efficient manner the potential effects on cognition in your early phase 1 trial.
Great. The next question we have is, how do you address the concern for a learning effect in driving or cognition testing?
The answer is real simple. It is related to training. When a subject participates in a study at Altasciences, the first thing they do is get screened for simulator sickness. That would determine if they're eligible to proceed. If they don't have that problem, then they go on to a very standardized familiarization drive where there's audio, video kind of presentation while they're in the simulator, taking them through the components. Then they do a screening drive where the operators are all trained on how to provide necessary training, coaching to the participants to get them to understand what the expectations are for them while driving, basically maintaining their lane position and controlling their speed. And we generally have demonstrated that we have, with the fairly simple driving task that we use for looking at sedating effect, that we have really good test-retest reliability and very minimal evidence of learning taking place after we've performed that training. For the cognitive testing, we also take people through training. Another way that we actually help reduce, practice/learning effects is when the subject returns to the unit after a washout period or for a check-in. We take them through kind of a familiarization drive and a cognitive test session just so they remain highly, fluent and capable and ready to perform the task.
Perfect. The next question includes, how are you sure that subjects are giving their best effort for these assessments?
I think, Gary...
Go ahead, Ingrid.
Yeah, I think Gary covered that pretty well in the last question, but also I think just to hone in on the actual training of the subjects themselves, but also of the staff. The staff are trained to recognize signs that a subject is potentially not giving their best effort, to remind them that, you know, they are performing the event, and to correct for anything that, you know, they're seeing. For the driving in particular, because it's such a long assessment, you know, you saw the video and some of the pictures. There's always an operator, so a trained operator in the room, you know, away from the subject so they don't get distracted by them. But they're taking notes. They're making sure that nothing, you know, funny is going on. And they're also there to, you know, to coach that participant and make sure that they are actually paying attention, doing their best, and correct for any, you know, instrument issue that may come up within the assessment. So it's really important to have that trained personnel that can recognize that something is going on that side. And Gary, would you like to add anything to that?
Yeah, I mean, it depends on what testing is being conducted. As you're well aware, performance testing is, you know, has the assumption that people are trying their best to perform on tests. And so we actually have some very sophisticated effort measures that can be applied, because we don't just measure speed, but we measure speed and accuracy, and we know quite a bit about how poor effort or lack of effort will be reflected in testing. And so that is something which we are keenly aware of, and do our best to get really valid data where that's not a problem.
Perfect. So the next question says, you mentioned that cognitive function should be assessed as early as phase 1 for new products. How is that handled, and are companies adhering to that portion of the guidelines?
Well, what do you think, Ingrid? As people are bringing you into first in human, how often are they doing that?
Yeah, so, you know, we talked a little bit about that during the presentation, but we didn't get too deep into it. But really, I think that really hasn't changed that much with the guidance coming out. I think developers of you know, novel CNS affecting drugs are already thinking about cognition. They're thinking about adding in some pretty tangible and descriptive measures to their first-in-human trials, and making sure that they're getting some good data as early as possible at different dose levels. And that's always been the case, and the better tools we have now, that helps with those assessments as well. What we still haven't seen a major shift in is other drugs where you may not expect to see a remarkable effect. Those clients are still, for the most part, opting to not include anything other than very simple, you know, things like objective questionnaires, how are you feeling, maybe some fast scales and descriptive questionnaires throughout the trial, but they're not in general bringing in that much cognition. We do recommend that at least some minimal testing be done for most compounds, and it really is just to de-risk the program, particularly when you get into the MAD, and there could be some concerns there.
And the reality is that with the tablet-based testing, it can be brought right to the patient's bedside if you're in a phase 1. That in fact you can administer a highly sensitive, repeatable measure that will look at things like processing speed, aspects of attention, and the total time needed would be, you know, under 15 minutes, and totally repeatable. And these are measures for which you know what the effect is of a lot of different drugs, maybe 20 different drugs on that particular tool, so that you can really gauge, you know, what is there any real meaning to the magnitude of that effect. So yeah, you may see a difference, but what is that? What does that compare to that really can inform you about the potential CNS effect?
Yeah, I would really. That's a great point. You know, those tests are very affordable to do. You don't have to do a huge analysis on them right away. It's not that different from, an early precision QT assessment where, you can take a look at the data later on even if you see a signal and do a more in-depth analysis. And it's not time-consuming and it's not expensive. It really makes a lot of sense to do with a validated platform like that.
Okay, that's great. And is there any major difference between regulators, for example, the EMA or FDA, for their expectations on when a dedicated driving assessment is required?
Good question, and I don't know that I know the answer, although we certainly are getting inquiries from Europe to help them conduct driving studies. I have not actually seen the EMA's guidance, or if they have actually published a guidance for driving. I'll ask Ingrid, do you know anything other than that?
No, what we've seen is that the expectations are very similar, and that the assessments that are required for the label are pretty much identical in thinking. So we haven't had to make any adjustments in the designs to address an agency.
Perfect. And how do you determine whether a population or healthy subject can be used to conduct a driving?
Well, I'd first refer back to the guidance, which basically requests that you include in your trial subjects who are similar to the population who will be taking this medication on a chronic basis. And so, one would then have to go to the next question you're to ask yourself, which is there a difference in driving for people who have this condition and healthy subjects? If the condition is not associated with any effect on driving, then in fact you could basically make a very good case for conducting the study in healthy subjects. Contrast to that, if you said, okay, well, this is a drug for treatment of, MS. MS is known to affect some aspects of processing speed by itself. Well, then that study should probably be conducted in patients with MS. And so it's really been, we've worked with the sponsors when they've asked us this question. We've, generally been able to resolve this to the satisfaction of the agency.
That's great. So I'm afraid that's all we have time for today, but we'll address all of the other questions offline. If you do have any other questions for Gary and Ingrid, then please feel free to send them to webinars@neuro-central.com. I would like to once again thank our wonderful presenters, Gary Kay and Ingrid Holmes, as well as you, our listeners, for your time and questions. If you would like to let us know your thoughts on today's webinar, please feel free to complete our feedback survey in the resource widget or tweet us at neuro_central. Don't forget to visit NeuroCentral at www.neuro-central.com for more webinars. Thank you again for attending, and goodbye.